Hey friends, Dr. Borole is here. Today we are going to learn the pressure sensors. Those are providing electrical output directly. Some of the pressure sensors provides direct electrical output are based on the following principles. The potentiometric sensors, the strain gauge based pressure sensors, the LVDT based pressure sensors and piezoelectric pressure sensors. Out of this, the potentiometric sensors, strain gauge sensors are the passive sensors, whereas LVDT based sensors and piezoelectric sensors provides the direct electrical output. LVDT based sensors has to feed with the primary supply, whereas piezoelectric sensors are the active sensors, they don't need any external power supply. The strain gauge sensors we have seen earlier under strain gauges. LVDT pressure sensor also studied under LVDT topic. The piezoelectric sensors, we have seen earlier the piezoelectric effect. Here we will revisit it. The piezoelectric pressure sensor figure is shown here. It consists of a piezoelectric crystal sandwiched between the two plates. It uses the piezoelectric property of the quartz crystal. When the pressure is applied to the piezoelectric crystal in y direction, say, then there will be the voltage developed in x-axis direction. And this happens because of change in the charge center of the molecules. We have seen this piezoelectric effect earlier in the lectures. When the pressure direction is changed, the direction of EMF induced is also changed. A typical piezoelectric sensor is shown here in the form of a disc. When the pressure is applied to this disc, the voltage will be induced and that is available at the output by the connecting wires. Inside this disc, there is a simple piezo wafer. Another configuration is cantilever configuration. The construction is typically shown here. One side is connected to the rigid support, and when the force is applied to this another end of the cantilever configuration, then there will be twisting force developed in the cantilever beam, and due to that there is a piezoelectricity generated, whereas compression type of piezoelectric crystal is shown in the first figure. The another type of pressure transducer is a capacitive transducer. The figure shows here a simple construction of a capacitive type of transducer. It consists of a metallic former body and the diaphragm is stretched between the two ends of the cavity. Under normal condition, this diaphragm is straight, shown by the dotted line. When the pressure is applied to the diaphragm gets deflected. And therefore, the distance between the diaphragm and the internal static plate is changed. This internal static plate and the conducting diaphragm is acting as a parallel plate capacitor. And therefore, capacitance of the capacitor is proportional to the distance between the plate. We know the formula for the capacitance. C is equal to epsilon A by D, where D is the distance of separation of the plates. So here pressure gives rise to change in the distance of separation, that gives rise to the proportional change in the capacitor. The another figure here shows a differential capacitor arrangement for measurement of differential pressure. Here this figure shows that P1 is applied from one port and P2 is applied from the another port. The diaphragm is stretched in between the two fixed plates. Whenever there is a difference between P1 and P2, the diaphragm will be either deflected left or deflected right. And therefore, the capacitance C1 between the left plate and the diaphragm will be different than the capacitance between the diaphragm and the right side plate. And this differential capacitance gives rise to the different voltages developed across these capacitors, that is E1 and E2. The external circuit reads the difference between the two, that is E1 minus E2. And that differential output is then proportional to a difference in the pressure. Such type of differential pressure transducer can be used for measurement of barometric pressure, that is pressure measurement with respect to atmosphere, or one of the pressure is maintained at absolute zero, then it is sealed with the vacuum and other port is applied with the pressure to be measured. So then this pressure will be the absolute pressure with respect to vacuum. In some application, we can measure the pressure P2 with respect to another pressure P1. Then this sensor provides the output that is proportional to the difference between the two pressures. It is possible to have a rotary capacitor also. One plate is fixed and another plate is movable. Here, when the plates are moving, the area of overlap will change and that will changes the capacitance between the plates. So rotary motion generated due to the deflection created by the pressure applied to the sensor gives rise to change in the capacitance. So rotary capacitor can also be used in some cases. The typical assembly, a typical practical assembly of the pressure sensor is as shown in the figure. It consists of a metallic diaphragm and it is hermetically sealed in a steel cabinet. And this can be used as a differential pressure device. That's all about the pressure sensors. The next sensor type we are going to learn is a biosensors. How biosensors are defined? Biosensors are basically a chemical sensing devices in which biologically derived recognition analyte is coupled to the transducer to measure the biochemical parameter of the analyte. Means whatever the biological sample that will be taken, that is called analyte, and that is reacting with a biochemical material of the sensor, which produces the response from the sensor and that can be measured by the transducer connected to the biochemical receptors. Typical parts of the biosensors are the biological element. This biological element is basically the component which recognize the analyte, often called as bioreceptors. These bioreceptors are normally the enzymes, or they can be antibodies, they can be cells, it can be nucleic acids, or it can be aptamers. There are various biochemical materials those can be used for detection of the various properties of analyte. The analyte can be a blood sample, it can be a urine sample, or it can be other samples taken from the body. The second part of the biosensor is a transducer component. Basically, it is a semiconductor material or nanomaterial component which will convert the response of biological element into electrical signal. This electrical signal can be connected to the electronic system that can signal condition it for the appropriate display purpose or recording purpose. The bioelements, also called as bioreceptors, uses biomolecules from organisms to interact with the analyte. The bioresponse of this interaction is then converted to electrical signal by the transducer. As per the particular applications, the biosensors are also known as immunosensors, electrodes, resonant mirrors, chemical canaries, biochips, glucometers, and biocomputers. The biosensors elements are as follow. The variety of substance may be used as bioelements in biosensors. These substances are nucleic acids, proteins, including enzymes, antibodies. Antibodies based biosensors are normally called as immunosensors. Plant protein or lectin, a complex material like tissue slices, microorganisms, and organelles can be used for detecting the various properties of analytes. A typical construction and the parts of biosensors are shown here. The biosensor consists of a transducer which basically detects the various signal generated by biorecognition layer. These biorecognition layer elements are shown here as biosensor elements. The chemical reaction takes place here. If you see, the various forms of sample molecules are shown by different shapes. The biorecognition element can only receive the particular type of element only, and it will receive that element, and there is a chemical reaction takes place, and output of that chemical reaction is then converted by the transducer to electrical signal. The electrical signal will be amplified. Maybe it can be controlled by microcontroller and displayed on the display, or it can be recorded on a printer. The biosensor block diagram is shown in the following figure. See, whatever the analyte that is called sample, maybe blood sample for example, it is given to a biosensor, that is basically a biocatalyst. It will convert that sample to product, and during this reaction there are some parameters of the product, those changes, and those changes will be detected by the transducer, and transducer produce the signal in the form of electrical parameter that can be further amplified if required and displayed. Some of the biosensor types are electrochemical biosensors, amperometry biosensors, potentiometric biosensors, physical biosensors, piezoelectric biosensors, optical biosensors, and wearable biosensors. Various applications of the biosensors are common healthcare checking areas, the biosensors are used. Metabolic measurement for various metabolic parameters, those can be measured using biosensors. For example, measurement of insulin quantity and insulin treatment areas, the biosensors can be used. The clinical psychotherapy and diagnostic of diseases where biosensor finds the applications. The drug improvement and offense detection areas also finds the application of biosensors. Ecological pollution control also finds the application of biosensors for detection of pollution. A typical example of a biosensor is blood glucose measurement sensor. The blood glucose biosensor is used to measure the glucose content in the blood of a diabetic patient. In this sensor, the enzyme glucose oxidase is used as a catalyst, and that generally the reaction with the blood took place, which makes the oxidation of D-glucose, means the content of the glucose in the blood. The reaction is shown below. Say D-glucose plus H2O plus O2, that gives rise to D-gluconic acid and H2O2. D-gluconic acid changes the pH value of the sample, and here the oxygen content will be liberated here. Those can be detected by using this glucose biosensor. The principle of glucose measurement is just a chemical reaction of the glucose oxidase with the glucose. It will takes place with the reduction in O2. It produces the H2O2 in the reaction, and there will be change in the pH value due to the gluconic acid. The measurement can be done by measuring the oxygen level by the two electrode. One electrode measures the ambient O2 level and other measures the O2 utilized during the reaction, and the difference of the O2 level is proportional to the glucose content in the blood. A typical blood glucose measurement strip is shown here. Here there are two electrodes. There is a exposure vent here, and there is a glucose oxidase gel. The blood sample is placed in this glucose oxidase. The chemical reaction takes place. The oxygen atoms are liberated, and those can be detected by these 2-point electrode cathodes. And the difference in the oxygen detected is proportional to the blood glucose present in the blood of diabetic patient. This way the biosensor works. That's all, friend, about the sensors. This ends unit number 5. Here there are some short answer questions and the long answer question for practice. So we will stop here. We will start unit number 6 in the next session. Till then, bye bye.